Hello, everyone. I thought I would read to you about Mr. Craig Venter. This isn't a fantasy look at the future. We're doing the future, he says. Uh, the pioneering American scientist who created the world's first synthetic life is building a gadget that could teletransport medicine and vaccines into our homes or to colonists in space. Craig Venter, this is from 2013. Craig Venter reclines in his chair, puts his feet up on the desk, and gently stroking his milk chocolate colored miniature poodle, Darwin, asleep in his arms, shares his vision of the household appliance of the future. It's a box attached to a computer that would receive DNA sequences over the internet to synthesize proteins, viruses, even living cells. It could, for example, fill a prescription for insulin, provide flu vaccine during a pandemic, or even produce phage viruses to targeted to fight antibiotic resistant bacteria. It could help future Martian colonists by giving them access to the vaccines, antibiotics, or personalized drugs they needed on the red planet. And should DNA based life ever be found there, a digital version could be transmitted back to Earth, where scientists could recreate the extraterrestrial organism using their own life printing box. We call it a digital biological converter. And we have the prototype, says Venter. I am visiting the office and labs of Venter's company, Synthetic Genomics Incorporated, in La Jolla, uh, a wealthy seaside enclave north of San Diego, California, where he also lives, because the pioneering American scientist dubbed biology's bad boy wants to talk about his new book released this week. The West Coast office of J. Craig Venter Institute has his not for profit research, whose research discoveries the company takes forward. It's a 5-minute drive away. Darwin's squeaky toys litter the floor, and Venter's wife, Heather Kowalski, who's also his publicist, sits within earshot, her legs tucked under her on a sofa. The walls are covered in awards, including his highest honor so far, his 2008 National Medal of Science Award, America's most prestigious scientific prize bestowed by the president. That's him getting the prize for his dedication to advancing the science of genomics, the study of genomes of organisms, and for his work in understanding and communicating its implications. The professional prizes are mixed in with sailing pictures and paraphernalia. The book, Venter's second, after his 2007 autobiography, is called Life at the Speed of Light: From the Double Helix to the Dawn of Digital Life. It looks at the future Venter is aiming to create through his scientific endeavors in synthetic biology, a kind of turbocharged version of genetic engineering where scientists design new biological systems, even synthetic life, rather than just tweaking existing organisms by inserting a gene here or there. In 2010, Venter grabbed the attention of headline writers and scientists around the world by announcing what he calls the world's first synthetic life. He took a synthetic bacterial genome constructed from the chemicals in the laboratory and, as Venter puts it, booted it up by inserting it into a living single-celled bacterium. The cell replicated itself into a colony of organisms containing only the synthetic DNA. It's like a whole new concept of life that, until our experiment, no one had had, he says. It was later reviewed by the Presidential Commission for the Study of Bioethical Issues, which gave it the green light and recommended self regulation by synthetic biologists. Interesting. Venter, who has a reputation for arrogance, uses his book to describe the nearly 15 years of scientific work that led up to his 2010 breakthrough. It also positions that work at the pinnacle of years of landmark discoveries by the biggest names in biology. A reader could be forgiven for thinking the book is really aimed at the Nobel Prize Committee, but Venter claims he just wants more people to understand him. One of the motivations for the book is to put this in a historical context because of all the confusion out there when we did it, he says. I think the work that we have done with the first genome in history and the human genome and with the first synthetic cell, is certainly one of the world caliber that obviously earns big prizes. Nobel Prizes are very special prizes, and it would be great to get one. The book is not a campaign to get one. Venter also wants people to know what's coming next, the futuristic home gadget he's building, and how it would allow what Venter calls biological teleportation. The way he sees it, making a digital copy of an organism's DNA in one place and sending the file to a device somewhere else that can recreate the original life form is not so different from the sci-fi ideal of teleportation, where matter moves from place to place in an instant in the style of beam me up, Scotty. It is just that it was never envisioned this way. Venter's isn't the only recent book by a leading scientist in the field to make wild claims about synthetic biology's future. Last year, George Church of Harvard proposed in his own book, Regenesis, that Neanderthals might be resurrected with the help of an extremely adventurous female human. But Venter, who dismisses Church's idea as fanciful in his book, tells me that his bold ideas are different because here in La Jolla they are becoming real. Mine is not a fantasy look at the future, says Venter. The goal isn't to imagine this stuff. We are the scientists actually doing this. Patrick McRae, a historian of science at the University of California, Santa Barbara, calls Venter a visioneer, a scientist who has not only a clear, big, and somewhat hubristic view of the future and his role in it, but the technological know-how to make it happen, along with the skills to bring money and people to their ideas. He's no armchair futurist, he says. Paul Fremont at Imperial College, London, admits he can't think of a scientist with a bigger ego, but believes it's important to have people like Venter who doesn't sit in the mainstream scientific establishment. He doesn't sit in the mainstream scientific establishment. It invigorates the rapidity of our development and makes public funders aware of things that they are perhaps a bit slow to be aware of, he says. John Craig Venter was born in 1946 in Salt Lake City, Utah, but grew up in San Francisco's Bay Area. After scraping through high school, he moved to Southern California to devote his life to the pleasures of surfing, only to be drafted into the Vietnam conflict. It was during those years that Venter, the drifter, gave way to Venter, the overachiever. Venter served in a Navy field hospital caring for wounded soldiers before returning to the U.S. to attend community college, transferring to the University of California, San Diego, where he received an undergraduate degree in biochemistry and a PhD in physiology and pharmacology. He believes he has the shortest PhD on record for the University of California, just about 3 years, and I published 21 scientific papers. Academic and research appointments followed, including at the U.S. National Institutes of Health, where Venter developed a new strategy for rapidly discovering genes. But being a cog in a larger machine rankled, and in 1992 he founded his own nonprofit research institute, the Institute for Genomic Research, later absorbed into JCVI. In 1995, Venter's team unveiled the first complete genome sequence of a free living organism, the bacterium H. influenza. It was made possible by a technique of Venter's invention for rapidly reading the code of DNA sequences called shotgun sequencing. Venter earned his bad boy tag in 1998 when he created a new company. I wonder if that's what these PCR tests are about. It's taking your DNA and getting the sequences so that they have all of our DNA on record, which they likely do already. Venter earned his bad boy tag in 1998 when he created a new company, Celera Genomics, to try and beat the publicly funded effort to becoming the first to sequence the human genome. The $5 billion public project was already 3 years in, but had an estimated 7 more to go. Venter said Celera would use shotgun sequencing and other new techniques to get the job done in only 3 years, with some in academia fearing Venter would be able to use patents to lock up access to vital information about human biology. The public project frantically reorganized and a scientific race ensued. Finally, after 3 years of very unfriendly competition, both efforts jointly announced that the genome had been mapped in 2000. Some of those people still hate me today, said Venter. I tipped the system over, and that 5 billion was going to be the gravy train for a lot of people for a very long time. He now dismisses the achievement that may yet earn him a Nobel Prize. It's just a 3-year diversion from his life's true work on making synthetic life. The first major breakthrough from that line of work came in 2003 when Venter's team made the simple virus X174 synthetically. In 2008, he synthesized the genome of a bacteria that infects the human urinary tract, Mycoplasma genitalium. Finally, in 2010, Venter announced that he had made the first synthetic bacterial cell. Venter is clearly still annoyed by the reception of his 2010 result received from some fellow scientists who argue that he had not created his life from scratch because he had used an existing natural cell to boot up the artificial genome. You see that statement over and over. But what does that even mean, Venter asked, using the analogy of baking a cake to try to dismiss his skeptics. A baker who starts with flour, sugar, and eggs gets credit for his creation, not accusations that he should have begun with atoms and molecules. Besides, he adds, just once the original cell reproduced, the question should be considered settled. We did rely on pre-existing life as the boot-up system for the chromosome, he says, but then it took over and started running the cell to its own specifications in what we call the synthetic cell. There is not a single molecule of the original form there. It's like converting you into a frog. Wow. We did rely on pre-existing life as the boot-up system for the chromosome, but then it took over and started running the cell to its own specifications. That is his synthetic cell. So if you don't believe that you can be rewritten by these vaccines, then you're deluding yourself. But I digress. Let's read on. Venter thinks that with the 2010 announcement, he finally answered the question posed by physicist Erwin Schrödinger in his 1944 book written for the lay reader, and of which Venter owns the first edition. What is life? Life is a DNA software program, says Venter. All living things are solely reducible to DNA and the cellular apparatus it uses to run on. The DNA software both creates and directs the more visible hardware of life, such as proteins and cells. With that question settled, says Venter, it's clear that if you give an organism a new software by rewriting its genome, then you have rewritten the software and life itself. He dismisses his scientific critics who say there's more to remaking life than creating DNA molecules as guilty of a kind of modern-day vitalism, the pre-scientific notion that an intangible something sets life apart from other things made from atoms and molecules. Although Venter works on single cells, he says he believes it holds true for even the most complex organisms. I can't explain consciousness yet, but like anything else, it will be explainable at the molecular level, the cellular level, and therefore the DNA coding level. You hear what he's saying? I can't explain consciousness yet, but I'm working on it, and he's going to it's going to be explainable at the DNA coding level, which they're planning on recoding you, us, you, me. Another common complaint about his 2010 work that it shows the scientist is intent on playing God. Venter seems happy to embrace, in the restricted sense that we had shown with this experiment how God was unnecessary for the creation of life. I suppose that we were, he writes. We now have a set of tools that never existed before to play creator, he says. Venter's current project, the Digital Biological Converter, or the DBC, is his attempt to put those tools into one convenient box. The current prototype is supported by the U.S. Department of Defense's research agency, DARPA, and is 8 feet long and 6 feet high. We have teams working to minimize it, to make it faster and more reliable, says Benner, who sees the devices which synthetic genomics will sell in hospital, workplaces, and homes. A major test of the prototype DBC's capabilities is planned before the end of the year. The current prototype can produce only DNA, not proteins or living cells, but even that could be enough to make the device practical. Some vaccines are made using DNA molecules, points out Venter. If there's a pandemic, everyone around you is dying, and you cannot go outdoors, you can download the vaccine in a couple of seconds from the internet, and that digital file will allow DBCs in homes, hospitals, and companies just to spit out a loaded syringe. His researchers believe their current prototype is already capable of producing DNA precisely enough that it could be used as a vaccine. Venter also sees a DNA printing version of his device helping with more regular medical care. It could print out the DNA that encodes the hormone insulin, so important to diabetics, he says. Adding that DNA to a protein synthesis kit, a tool that is commonplace in research labs around the world, would produce the finished treatment for injection. Venter also has the antibiotics crisis in his sight. Before penicillin, it was common in Russia and Eastern Europe to treat some infections using naturally occurring viruses known as bacteriophages or phages that killed the bacteria responsible. The DBC could bring back so-called phage therapy and make it more effective, says Venter. The future will be that if you have a bacterial infection, you quickly get its genome sequenced. That will take minutes, and in a very short period of time, we could design a phage that would attack that bacteria very specifically, he says. Because of the way phages attack their bacteria victims, making just the DNA of one person it needs is enough, says Venter. The DNA is the drug that kills the bacteria. Looking further ahead, Venter intends DBCs to print living cells using an automated and improved version of the process behind his 2010 breakthrough synthetic cell. Work on that is currently underway with the focus on creating what he calls the universal recipient cell, a kind of biological blank slate able to receive any synthetic genome and come to life. We have to engineer it, but we think it's doable, says Venter. It would mean the possibility of ready access to new cells, which currently Venter and other arms of his work, along with others, is currently trying to engineer to produce necessities such as therapeutics, food, and clean water. Their genomes could be made available to be downloaded and printed on DBCs around the world. In parallel to the development of the DBC, Venter scientists are also working on a machine called the digitized life sending unit, intended to complete his vision of an end-to-end system for biological teleportation. The sending unit's job is to sample robotically, sequence a genome from the sample, and generate a digital DNA file that is then sent to a DBC to recreate the original life in a new location. It's a project that has attracted support of NASA, which doubtless hopes that the future robotic Mars missions could pack a all right, I've given up on that. Life sending unit, digitized. I don't know why that kept giving me tongue tying me. Life sending unit so that any Martian microbes discovered could have digital copies of their genome sent back to Earth. No prototype of the device is yet ready, but Venter says JCVI scientists working in California's Mojave Desert trying to work out how a robot could autonomously isolate microbes from soil and sequence their DNA. Venter believes that scientists using DBC on Earth equipped with a universal recipient cell could then recreate a Martian organism in a maximally contained laboratory. Having alien species being beamed back here and being recreated does sound like science fiction, but it's potentially real, says Venter, who argues that approach would be far cheaper than trying to bring back a sample back to Earth and come with much less of a risk of contaminating our planet. Venter believes that any humans that make the trip to the Red Planet will be packing one of the DBCs, enabling them to receive supplies such as vaccines, antibiotics, and cells that become new food sources. The distances are too great to do it any other way, he says. My friend Elon Musk, and others are determined to colonize Mars in the not too distant future, he says, dropping the name of the billionaire founder of PayPal, who leads both electric car maker Tesla and private rocket company SpaceX. As to whether we will ever be biologically teleporting humans, Venter gives the notion short shrift. That is not going to happen anytime that we know of. There are two different worlds and two different scales, and the science isn't anywhere remotely supportive of that any more than it is recreating a Neanderthal with a willing woman. Venter believes he's able to envision and ultimately achieve such big goals because he's escaped the narrow-mindedness of academic science. He thinks the scientific establishment would get more done if it adopted his way of doing things. The trouble is, the field of science, medicine, universities, biotech companies, you name it, have been so splintered, layers, subdivided, hacked, that people spend their entire career studying one tiny little cog of life, he says. If I could change the science system, my prescription for changing the whole thing would be organizing it around big goals and building teams to do it. That's what we do. I have created a team science versus the university system with 200 prima donnas, each of their own little space. Interview over, we leave Synthetic Genomics main building and we take a short drive in Venter's 1996 Aston Martin. Venter collects cars and motorbikes, though the Synthetic Genomics campus and through the genomics campus and pass down his algae greenhouses where Venter scientists are experimenting with the cells of the future to our destination, Venter's new DNA synthesis factory. The dog Darwin, who's traveled down separately with Kowalski, also joins us for the tour. Inside the gleaming building, only 6 months old, white-coated technicians work with liquid handling robots with which pipette chemicals and enzymes to fill customers' orders for pieces of synthetic DNA. Pharmaceutical and agricultural companies are the main ones so far. Other companies also synthesize DNA commercially, but Venter's is making the largest and most complex. Then someone mentions the DBC. Should I show it to her? We have part of it here, one of the lab managers asked Venter. Looks are exchanged, however, it's clearly not in Venter's master plan for today. America faces an epic choice. In the coming weeks, and the results will define the country for a generation. These are perilous times. Over the last 4 years, much of what the Guardian holds dear has been threatened. Well, that's sorry. I don't know why I didn't realize that's their plea to help. This is from the Guardian. But I just thought I would read that article. Did you catch that? Venter is clearly annoyed by the reception in his 2010 result received from some fellow scientists who argue that he had not created his life from scratch because he'd used an existing natural cell to boot up the artificial genome. You see that statement over and over. What does that even mean, Venter asked, using the analogy of baking a cake to try to dismiss his skeptics. A baker who starts with flour, sugar, and eggs gets credit for his creation, not accusations that he should have begun with atoms and molecules. Besides, he adds, once the original cell reproduced, the question should be considered settled. We did rely on pre-existing life as the boot-up system for the chromosome, he says, but then it took over and started running the cell to its own specifications. In what we call the synthetic cell, there is not a single molecule of the original from there. It is like converting you into a frog. So once they boot this messenger RNA up into you, the internet, and start running their code, it's going to take over, literally rewriting your DNA, making a synthetic creature with not a molecule left of the original DNA. But I have some other links here about Venture. He's making biofuels for this brave new world. Algae. And let's not forget Cynthia. And he brags that his scientists, they watermark their creations. So I shared a video called SynBio, and there's a link in there that you should watch about Craig Venter. It's from 2010. Let's see. New fuel, life, synthetic life could help provide 21st century society with new fuels, medicines, food, and nutritional products, supplies of clean water, and other resources. Venter, a pioneer in the field, led the team Celera Genomics that went head-to-head with the government and foundation-funded Human Genome Project. Okay, let's get down here to see. Okay. A project at SGI, for instance, aims to engineer algae cells to capture carbon dioxide and use it as a raw material for producing new fuels. He also described his work in sequencing the first draft human genome in 2001. And his team at Celera Genomics, as well as the work on his complete diploid genome published in 2007 at JCVI, along with collaborators at the Hospital for Sick Children in Toronto and the University of California, San Diego, in addition to continued analysis of Venter's genome. So, and he has this company, Synthetic Genomics. Let's see. Sorry, my computer's going kind of slow. Advancing genomics to better life, bringing algae from lab to field. I wonder if it says anything about Cynthia on here on this website. Anyways, I just wanted to share the article about Craig Venter. When you think about what's going on today with the COVID and what they're about to roll out with synthetic creation, where they're talking about you at some point, there'll be a literal beam me up Scotty type deal. But none of this talks about the spirit or the soul. He's a pretty arrogant man. Anyways, let me know what you guys think. Thanks for listening.